Hello everyone, a very welcome to Vyas Certification Kota. So today in the series of NCERT discussion, we are going to discuss another chapter from class 11th, that is chapter number 8, Cell, the Unit of Life. So let's discuss the NCERT questions of this chapter, and the very first question on the screen is, which of the following is not correct? Option A is given as Robert Brown discovered the cell. Second option we have given is Schleiden and Schwann formulated the cell theory. Then the third one we have is Virchow explained that cells are formed from pre-existing cells, and the last option we have is a unicellular organism carries out its life activities within a single cell. So in this question we have to find out which one is not correct, right? So as we know that the cell theory was actually formulated by two scientists named as Schleiden and Schwann. Then the third statement you can see, Virchow explained that the cells are formed from pre-existing cells only. This is also a correct statement, and the fourth option is also correct. We have left with only one option, that is option A, which is not correct. As we know that cells are not discovered by Robert Brown, right? The cells was actually discovered by another scientist named as Robert Hooke. Alright, so cells were discovered by another scientist named as Robert Hooke. He discovered the cells in the year of 1665. He observed dead cells of plant, or we can say cork, right? And from that he discovered cell. He resembled or remarked the structure of cork with cellular or little rooms, and that's how the discovery of cell has been taken place. So option number 1 is actually the answer because it is not correct. So we will writing the answer A as option A. Option A is not correct. So we are going to correct this. So we know that Robert Hooke discovered the cells in the year 1665. It's 5, right? So Robert Hooke actually discovered the cells in the year of 1665. How he observed? He actually observed the dead cells of cork, and he remarked the structure of that as cellular or little rooms, right? So option number A is not correct, as we know that Robert Brown was not the scientist who discovered cells. Actually the cell were discovered by Robert Hooke. So this is the complete answer of question number 1. Now we have next question on the screen, that is question number 2, and this question is asking new cells generate from. Option number A is bacterial fermentation. Option number B is regenerating of old cells. Option number C we have is pre-existing cells, and the option 4 we have is abiotic materials. So we know that whenever we talk about arising of new cells, they always arise from pre-existing cells, right? According to the theory of biogenesis, always remember that new cells arises from pre-existing cells, right? So option number C is the correct answer for question number 2. Option number C is the correct. We know that according to the theory of biogenesis, the new cells always arise from the pre-existing cells, pre-existing cells. According to the theory of biogenesis, always the new cells are going to arise from the pre-existing cells only. So this is the complete answer of question number 2. Now let's discuss next question, that is question number 3, and in this question number 3 we have to match the following terms. The terms given in the column number 1 with the terms given in the column number 2. So let's see, the A option we have given is cristae, the option number B we have given is cisternae, and the third we have is thylakoids. And in the column number 2 we have flat membranous sac in stroma, enfoldings in mitochondria, disc-shaped sacs in Golgi apparatus. So we have to match them. We have to match them with the column number 2. So if we talk about the first one, that is cristae. You all know that what is cristae. If we talk about mitochondria, mitochondria is double membranous organelle. If we talk about the inner membrane of mitochondria, we know that the inner membrane of mitochondria folds, right, folds inwardly to increase the surface area for respiration. So what is cristae? When the inner membrane of mitochondria folds inwardly, we call it as cristae. So A will go with this enfoldings in mitochondria. A will go with enfoldings of mitochondria. Then we have cisternae. We have cisternae. You all must be knowing about the structure of Golgi apparatus, right? So whenever we are talking about the structure of Golgi apparatus, we know that the long sac-like structures in the Golgi apparatus, which is a cell organelle in the cell, right? Those sac-like structures in the Golgi apparatus are nothing but we call them as cisternae. So all these cisternae will go with disc-shaped sacs in the Golgi apparatus. Now we are left with thylakoid. So obviously thylakoid will go with option number 1, but you must know what is thylakoid. We know that chloroplast, right? Chloroplast, which is present or which is found in only plant cells. That cell organelle such as chloroplast is not found in animal cells. If we talk about chloroplast, then if we talk about chloroplast, we know that there are thylakoids, there are grana, right? Thylakoids are coin-like structures. Thylakoids are coin-like structures which bear or which contain pigment, chlorophyll pigment, which is helpful in the process of photosynthesis. So what are thylakoids? First of all, it is present in the chloroplast, which is a cell organelle found in the plant cells only, right? So thylakoids are nothing but the flat membranous sacs in the stroma, right? Stroma of chloroplast, stroma of chloroplast. So this is the answer, that thylakoid will go with option number 1. So we are going to write C, and we have matched all the terms given in the column number 1 with the terms given in the column number 2. So this is the complete answer of question number 3. Now let's come towards the next question, that is question number 4, and this question is asking we have to select first of all which of the following is correct. So we have to select the correct statement, right? We have to select the correct statement, and we have to choose among these options. One statement is correct and all other are actually wrong. So option A or statement A is saying that cells of all living organisms have a nucleus. Then option B or statement B we have is both animal and plant cells have a well-defined cell wall. Then we have option number C or statement C, in prokaryotes there are no membrane bound organelles. Then the last statement we have is cells are formed from abiotic material. Alright, we know that we can easily find out the correct statement among the following given following options or statements. The option number A is incorrect, right? Then if you talk about the option B, which is also incorrect, as animal cells do not contain cell wall, right? We do not have cell wall. Then the option number C, in prokaryotes there are no membrane bound cell organelles are present. That is very much correct answer. That is the correct statement. Option number C is the correct answer, as we know that in prokaryotes or in prokaryotic cell you are not going to find any cell membrane, any cell organelle which is a membrane bound, right? This is a primitive type of cell which lacks all membrane bound cell organelles. So this statement is the correct statement. Option 4 or 4th statement is also incorrect, as we know that always cells are going to form from the pre-existing cells, right? So option number C is the correct answer, as we know that there are no membrane bound cell organelles are going to be found in the prokaryotic cells or prokaryotic organisms. So this is the complete answer of question number 4. Now let's discuss question number 5. So question number 5 is asking what is mesosome in prokaryotic cells. Also we have to mention the functions of mesosome. So first of all, what is mesosome? Mesosome are convoluted membranous structures found in the prokaryotic cells and are formed by the invaginations of plasma membrane. Right? What are mesosomes? Mesosomes are nothing but the membranous structures, convoluted membranous structures which are formed by the invaginations of plasma membrane in the prokaryotic cells only. Right? This is mesosome. Now let's talk about the functions of mesosome. So mesosome helps in the formation of plasma membrane. It helps in the formation of cell membrane. It also helps in the DNA replication and protein synthesis. Right, mesosome helps in the formation of cell membrane, and then it also helps in the DNA replication and protein synthesis also. Also this extensions or mesosomes helps in the process of secretion as well as bacterial respiration. Last but not least, it also increases the surface area of plasma membrane to perform various kind of enzymatic activities in the prokaryotic cells. So this is the function of mesosomes. Now let's write this answer. So we are going to write this answer like this. First of all, we are going to write what is mesosome. So start with mesosome. Mesosomes are convoluted membranous structures. Membranous structures found in the prokaryotic cells, found in the prokaryotic cells. Formed from the invaginations of plasma membrane. Plasma membrane. Now let's write the functions of mesosome. So we are going to write the functions of mesosomes. So the first function, the very first function of mesosome is it helps in the formation of cell membrane or plasma membrane. So it helps in the formation of cell membrane, DNA replication and protein synthesis and protein synthesis. Right? The next function of mesosome or these extensions, as we know that these are nothing but the extensions of plasma membrane in the prokaryotic cells. So the first function of these mesosomes are they are helping in the formation of cell membrane, they are helping in the DNA replication, as well as they are helping in the formation of proteins. Then the next, if we talk about the function of mesosomes, then it helps in the equal distribution of chromosomes into daughter cells, into daughter cells during cytokinesis, the completion of cell division. Then if we talk about the third function, it increases the surface area of plasma membrane to perform various enzymatic activities, Last but not least, they are also helpful. It helps in secretion and bacterial respiration. It also helps in the secretion as well as bacterial respiration. So all these are the functions, important functions of mesosomes. Remember that mesosomes are going to be found in only prokaryotic cells. They are not going to be found in eukaryotic cells. For various kind of functions mentioned on the screen, we have different kind of organelles in the eukaryotic cells. But as we know that prokaryotic cells are the primitive type of cells and they do not contain any kind of membrane bound cell organelles. So they have mesosomes as the extension of their plasma membrane, which performs all these crucial functions, important functions for them, right? So this is the complete answer of question number 5. Now moving on to the next question, that is question number 6, and this question is asking, how do neutral solutes move across the plasma membrane? How do neutral solutes move across the plasma membrane? Also this question is asking that, can the polar molecules also move across it in the same way? If not, then how they transported across the membrane? So first of all, we are talking about how do neutral solutes, right? Before moving on to the how these neutral solutes or these polar molecules are going to move across the plasma membrane or cell membrane, first we have to know that what is cell membrane or plasma membrane. Cell membrane or plasma membrane is the outermost covering of cell. We know that cell membrane is the outermost covering of cell which separates the cell from its environment, right? This cell membrane allows some substances to move across the plasma membrane and it restricts the entry of some materials across the membrane. Right, this is why we call the cell membrane as selectively permeable, because it is allowing some substances to move across and it restricts the movement of some other material or some other substances. That is why the cell membrane is known as selectively permeable in nature, right? Now, this is what we know that about cell membrane. Now, if we talk about the movement of neutral solutes across the cell membrane, so neutral solutes will move across the cell membrane with the help of simple passive diffusion, right? They are absolutely going to move across the cell membrane with the help of simple passive diffusion. Now, what is diffusion? Diffusion is a process by which the molecules are going to be moved from the region of higher concentration to the region of lower concentration. This is what we call diffusion. So, by simple passive diffusion, neutral molecules or neutral solutes are going to be moved across the plasma membrane. But in case of polar molecules, they are not going to move across the plasma membrane with the help of simple diffusion, as we know that cell membrane is made up of bilipid layer, biphospholipid layer, right? Or we can call that the cell membrane is actually made up of phospholipid bilayer and some proteins, right? So the cell membrane is actually non-polar, and we are trying to move the polar molecules across it. So if we talk about the movement of polar molecules across the non-polar cell membrane, Then they require some special carrier proteins. They require some special carrier proteins to move across the plasma membrane. These carrier proteins are integral proteins which have a certain affinity towards some kind of solutes or some kind of molecules. And this is how, with the help of some special carrier proteins, some polar molecules can move across the plasma membrane. So we know that the solutes, the neutral solutes are going to be moved across the plasma membrane with the simple diffusion, whereas the polar molecules require special carrier proteins to move across the cell membrane. Right, this is the answer. Now let's write answer of question number 6. That first of all we are going to write about cell membrane. So cell membrane is the outermost covering of cell which separates the cell from its environment, Now, this cell membrane allows the movement of some substances in or out from the cell, while it restricts the movement of some other material, Hence, it is called as selectively permeable membrane. The cell membrane is actually selectively permeable membrane in nature because it allows some material to move in or out from the cell, and it restricts the movement of some other material. That is why this is actually semi-permeable membrane. Now let's talk about how do neutral solutes actually move across the plasma membrane. So neutral solutes move across the plasma membrane with simple passive diffusion. I hope you all know what is diffusion. If no, that I am going to write the simple definition of diffusion. Diffusion is the movement of molecules from the region of higher concentration to the region of lower concentration. Right? So we know that whenever we talk about the movement of neutral solutes, they are going to move across the plasma membrane with simple diffusion, with simple passive diffusion, right? But what about the movement of polar molecules across the plasma membrane? We know that they require some special carrier proteins, So let's write about the polar molecules movement. So the movement of polar molecules requires some special carrier proteins as the cell membrane is made up of phospholipid bilayer and proteins. These special carrier proteins are nothing but the integral proteins located in the plasma membrane only. So these special carrier proteins are the integral proteins right? These carrier proteins are the integral proteins which helps or which have certain affinity, for some substances, and they help them to move across the plasma membrane. Right? This is how we know the movement of polar molecules across the plasma membrane takes place with the help of carrier proteins. And if you talk about the neutral solutes, they are going to move across the cell membrane or plasma membrane with the simple diffusion, right? So this is the complete solution of this question. Now let's discuss question number 7, and this question is asking name 2 cell organelles that are double membrane bound. What are the characteristics of these 2 cell organelles? Also we have to state their functions and we have to draw some diagrams of both of them. So first of all, if you talk about what are these 2 cell organelles which are double membrane bound? Alright, so the cell organelles which are double membrane bound we have are chloroplast and mitochondria. Right? So mitochondria and chloroplast are the 2 cell organelles which are double membrane bound. That means they have 2 membranes. They are covered with the help of 2 membranes. That is why we are calling them as double membrane bound structures or double membrane bound cell organelles. Let's discuss the mitochondria first. So what is mitochondria? Mitochondria is first of all the double membrane bound cell organelle or double membrane bound structure, which is divided into 2 aqueous compartments with the help of 2 membranes, that is outer membrane and inner membrane. The outer membrane of mitochondria or the outer compartment of mitochondria are very porous, that means they contain several cell organelles in them, whereas the inner membrane of mitochondria folds inwardly, right? So inner membrane do not have any cell organelle and the outer membrane of mitochondria have cell organelle. Now the foldings of inner membrane of mitochondria are known as cristae. The foldings of inner membrane of mitochondria is known as cristae, which increases the surface area of the cell. These cristae increases the surface area of cell, and this is the site, the inner membrane of the mitochondria is the site for ATP generating chemical reactions. Also, it contains various kind of enzymes, specific enzymes, and thus it is the site for aerobic respiration, right? The mitochondria also contain their own DNA and ribosome, and hence they are capable of making their own proteins. That is why we call the mitochondria as semi-autonomous organelle. This is about mitochondria. Now let's talk about the characteristic of chloroplast, which is also a double membrane bound structure or double membrane bound cell organelle. But that is very important, that you are not going to find the chloroplast in animal cells, right? Mitochondria is the cell organelle which is double membrane bound. You will find this mitochondria in the plant cell as well as in the animal cell. But the chloroplasts are not going to be found in the animal cells, and they are always found in the plant cells, right? Now let's come towards the characteristics of chloroplast. Chloroplast is also a double membrane bound cell organelle or structure which is divided into two compartments or two regions, the outer region and the inner region. It is also having two membranes, the outer membrane and the inner membrane, right? Now if we talk about the chloroplast, the two regions of chloroplast are grana and stroma. The grana are the stalks of flattened disc-shaped structures which are known as thylakoid. These thylakoid contains chlorophyll, which is a pigment used for photosynthesis, right? Now, if we talk about these granas, the thylakoids of adjacent granas are connected with the help of membranous tubules known as stroma lamellae. Now, let's talk about the next region of chloroplast, that is stroma. Stroma is a homogeneous mixture of chloroplast in which the granas are actually embedded. Also, there are various kind of enzymes are residing in the stroma of chloroplast which are helpful in the formation of carbohydrates and proteins. These are the characteristics of chloroplast and mitochondria. Now, we know that we have to mention their functions. We have to mention the functions of chloroplast and mitochondria. So, let's talk about the functions of mitochondria first. What are the functions of mitochondria? This helps in the formation of ATP, which is the energy currency of cell, which helps in performing all the vital activities of the cell, right? It also provides a site for aerobic respiration or cellular respiration, right? So, what is the function of mitochondria? Mitochondria helps in the formation of ATP, right? It also provides a site for cellular respiration. Mitochondria have their own DNA and protein, so they are also capable of producing their own protein. Hence, we are calling them as semi-autonomous. Also, mitochondria have specific enzymes which help in the formation of fatty acids, steroids and some amino acids. So, this is the function of mitochondria. Now, let's discuss the function of chloroplast. Now, we know that the chloroplast is divided into two regions, right? The one is grana and the another one we have is stroma. The grana are nothing but the stalks of disc-shaped like structures, that is thylakoid. Thylakoid contains chlorophyll pigment, which is very, very important in the formation of food by the plants. So, what is the function of chloroplast? It traps the sunlight, and uses the sunlight in the formation of food by the plants, right? So, this is the function of chloroplast. Also, the stroma of chloroplast have certain enzymes which are very helpful in the process of formation of carbohydrates and proteins. So, these are the functions of chloroplast and mitochondria. Now, we are going to write the answer of question number 7. Also, we are going to make the diagrams of chloroplast and mitochondria as well. So, let's start writing the answer of question number 7. So, first of all, we are going to write the very first answer, that is the two cell organelles which are double membrane bound. We know that the two cell organelles which are double membrane bound are the two cell organelles which are double membrane bound. Double membrane bound are mitochondria and chloroplast. Mitochondria and chloroplast, right? Now, let's discuss the very first organelle, that is mitochondria. Mitochondria. We are going to write about mitochondria. So, what is mitochondria? It is double membrane bound. Double membrane bound cell organelle which is divided into two membranes, the inner and outer membrane, inner and outer membrane, distinctly dividing two aqueous compartments. The outer compartment and the inner compartment. The inner membrane, the inner membrane of mitochondria is very porous. That means contains cell organelle, and the inner membrane folds inwardly, right, which is not containing any cell organelle but involves in the formation of ATP or involves in the site, in providing the site for respiration of cell, right. So this is mitochondria. Now let's talk about the inner membrane. We have a special name for inner membrane of mitochondria, that is cristae. So the inner membrane of mitochondria is known as cristae, right. The inner membrane of mitochondria is known as cristae. This is the site of aerobic, this is the site of ATP generating chemical reactions. Also it is a site for aerobic respiration as well, but we are going to write like this: the inner membrane of mitochondria is known as cristae and this is the site for ATP generating chemical reaction. This also contains, the inner membrane also contains specific enzymes, for aerobic respiration, right. So the inner membrane of mitochondria is actually involved in the ATP generating chemical reactions. Also it provides the site for aerobic respiration of cell. Now this contains its own DNA. We are talking about mitochondria, own DNA and ribosome. Thus can make its own proteins, hence they are semi-autonomous, right? These are the characteristics of mitochondria. Now let's write the functions of mitochondria. Also we are going to draw a diagram of mitochondria. So first we are going to write about the functions. So the first function of mitochondria is this provides the energy in the form of ATP, which is the energy currency of our cell. And with this ATP or energy, all the vital activities of cell are going to be performed, right? So this mitochondria provides energy in the form of ATP. Also, it provides the site for cellular respiration, right? So these are the functions. Now let's make a diagram to show how actually a mitochondria is. So we are going to make mitochondria like this. This is the outer membrane and these are the inner membrane. Right, this is the outer membrane. This is the inner membrane. Sorry, this is the matrix. And this is the inner membrane. Now, inside the matrix, they have their own DNA. Also ribosomes. So these are ribosomes. These are DNA. Right, so this is the labeled diagram of mitochondria. Now let's talk about another organelle, double membrane organelle, that is chloroplast. So we are going to write chloroplast here. Chloroplast is also a double membrane bound cell organelle. Right? So chloroplast is also a double membrane bound cell organelle which is divided into inner and outer membrane. Further divided into two aqueous compartments, that is grana and thylakoid. So what are grana? Grana are the stalks. These are the stalks of flat disc shaped structures known as thylakoids, which contains chlorophyll, Now, the thylakoids of adjacent grana are joined together with membranous tubule, known as stroma lamellae. Right? The next region of chloroplast is stroma. This is the homogeneous mixture. In which the grana are embedded. Right? So stroma is the homogeneous mixture in which the granas are embedded. Also, the stroma contains certain enzymes or specific enzymes. Also, it contains specific enzymes helping in the formation of carbohydrates and proteins, right? So this is what we have, the characteristics of chloroplast. Now we are going to discuss, or we are going to write the functions of chloroplast, and we are going to make the diagram of chloroplast, right? So let's write first the functions of chloroplast. Functions of chloroplast is they trap the sunlight energy and make food by plant. That means it involves in photosynthesis. Also, they help in the formation of carbohydrates and proteins. So this is the function of your chloroplast. Now we are going to make the diagram of chloroplast here, the diagrammatic representation of chloroplast. As we know that this is the double membrane bound structure, we are going to make the outer membrane, then the inner membrane. We know that this is the outer membrane. This one is inner membrane. Then this is the stroma, and inside the stroma we have grana, which is made up of disc-like structures known as thylakoids. These are thylakoids which are arranged one upon each other and making a stalk-like structure known as grana. So these are grana. Now we know that each thylakoid in the grana of each thylakoid in the adjacent grana are connected with the help of a membranous tubule known as stroma lamellae. So these thylakoids of the adjacent grana are connected to each other with the help of this membranous tubule known as stroma lamella. So first of all, this is stroma, right? This is stroma. This is intermembrane space. This is intermembrane space. And these are grana. These are grana. This whole thing is grana. Each disc-like structure in a grana. Each disc-like structure in a grana is thylakoid. Right? Now, these membranous tubules which are joining the thylakoids of adjacent grana are stroma lamellae. So these are stroma lamellae. Alright, so this is the complete labeled diagram of chloroplast, and with this we have completed the solution of this question. So let's discuss next question, that is, what are the characteristics of prokaryotic cell? So we have to give some characteristics of prokaryotic cell. First of all, what is prokaryotic cell? Prokaryotic cells are the primitive kind of cells, the primitive kind of cells. Now, as they are primitive kind of cells, they do not contain any kind of membrane bound cell organelles. There are no membrane bound cell organelles are present in the prokaryotic cells. They are primitive cells, right? If you talk about some examples, which kind of organisms do possess these prokaryotic cells or primitive cells? Bacterias. Bacterias are the prokaryotic organisms. Why they are known as prokaryotic organisms? Because they are made up of these prokaryotic type of or primitive type of cells. Bacterias or cyanobacterias do not contain any membrane bound cell organelles in them. Right, this is what we know that what is prokaryotic cell or prokaryotic organisms. Now, if we discuss the characteristics of prokaryotic cell, so they do not contain, the first characteristic of prokaryotic cell is they do not contain any membrane bound cell organelle in them. They do not contain any membrane bound cell organelle in them. They possess single circular DNA as their genetic material. They possess only single circular DNA as their genetic material along with the presence of plasmid. What is plasmid? Extra genetic material present in the bacteria, right? This is extra genetic material which is known as plasmid and which is present in only bacteria. Now, these are the two characteristics of prokaryotic cell, that is, they do not contain any membrane bound cell organelles, and also they possess single circular DNA as their genetic material along with the presence of plasmid. Also, they do not have mitochondria, as we know that they do not contain any cell organelle or which is membrane bound, so they do not contain mitochondria. Instead of that, they have mesosomes. Mesosomes are the extensions or invaginations of plasma membrane. So bacterial cell or any prokaryotic cell is having mesosomes as the extension or invaginations of cell membrane or plasma membrane, which helps in the process of respiration of them. Respiration of cell is actually done by the mesosomes and not by the mitochondria, because it is not present in the prokaryotic cell, right? So these are the some characteristics of prokaryotic cells. So let's write answer of question number 8. First of all, prokaryotic cells. Prokaryotic cells are the primitive type of cell which do not contain any membrane bound cell organelle. For example, bacteria, cyanobacteria. These are the examples of prokaryotic organisms. Let's write the characteristics. Of prokaryotic cells. Characteristics of prokaryotic cells. So the first characteristic of prokaryotic cell is they do not possess any membrane bound cell organelle, right? Next is they contain or possess only single circular DNA as their genetic material. Along with plasmid. This plasmid is the extra genetic substance in the prokaryotic cell, which we also use in the biotechnology, right? So this plasmid is the extra genetic material found in the prokaryotic cells or bacteria, which is used in the biotechnology processes. Then the next characteristic of prokaryotic cell is they have mesosomes, which are the extensions or invaginations of the cell membrane which helps in the respiration. So they have mesosomes for respiration process, right? So these are some characteristics of prokaryotic cells, and with this we have done the solution of this question number 8. Now let's talk about the next question, that is question number 9. Multicellular organisms have division of labor. Explain. So first of all, what are multicellular organisms, or what are the examples of multicellular organisms? We are the examples of multicellular organisms. Plants, animals, various kinds of organisms are the examples of multicellular organisms. What are actually multicellular organisms? Multicellular organisms are the organisms which are made up of millions or trillions of cells, right? That is why we are calling them as multicellular organisms. Now, what is multicellular organism? Simply, multicellular organisms are the organisms which are made up of millions or trillions of cells. Now, if we talk about what is division of labor, let's come towards this point. So, any multicellular organism having tissue. What is tissue? A group of cells. So, various kind of specialized cells performing similar function are grouped together as tissue in the body, right? Various kind of specialized cells performing a similar function, they are grouped together as tissue in the body, which are performing a specific function, right? This is tissue. Now, similarly, various tissues or various kind of tissues performing various kind of functions. This is known as division of labor in a multicellular organisms. In a multicellular organism's body, we have different kind of tissues at a definite place in the body. And these tissues at the definite place in the body are performing different kind of their specific functions. And this is what we call the division of labor in multicellular organisms, which cannot be seen in the unicellular organism, as only one cell is performing all the vital activities, all the functions of the cell or organism in that case, right? So this is what we have, the division of labor in case of multicellular organisms. Now let's write the answer of this question. Answer number 9. First of all, multicellular organisms are made up of millions or trillions of cells. Millions or trillions of cells, right? Now, the specialized cells performing a similar function. The specialized cells, the different specialized cells performing a similar function are grouped together as tissues in the body. Similarly, different or various tissues, which are nothing but the group of cells, so various tissues or different tissues at a definite place in the body are performing different specific functions. This is known as division of labor. In any multicellular organism. As we know that the division of labor is actually found in the multicellular organisms only. It cannot be found in unicellular organisms, right? So this is the complete solution of question number 9. Now coming on to the next question, that is question number 10, and this question is saying cell is the basic unit of life. Discuss in brief. Yes, cell is the basic unit of life. Cell is the fundamental basic unit of life. So what is the answer of this, or we have to discuss it in brief? So cell is the basic unit of life performing all the biochemical reactions or processes in order to live, right? So cell is the basic fundamental unit of life or basic unit of life. Now the need for all the living organisms to survive are same. They need to respire, they need to digest the food, they need energy, they also need to get rid of metabolic waste. So cells are capable of doing all the metabolic activities like these, right? That is why we are calling the cells as the basic unit of life or basic fundamental unit of life, right? This is the answer of this question. Now let's write the answer number 10. Cell is the unit of life. Cell is the basic unit of life performing the various biochemical processes or reactions right? Cell is the basic unit, cell is the unit of life performing all the biochemical processes of life in order to live. Now the basic needs of all living organisms to survive are actually same. We know that are same. They need to respire. They also need to digest food. They also need energy, and they also need to get rid of metabolic waste. Right? These are the basic needs of all the living organisms on the earth. Now, the cells are capable of doing or performing all the metabolic activities. All these are the metabolic activities. The energy they need, the food they want to digest, they want to respire, all these are the metabolic activities. So cells are actually capable of doing all the metabolic activities, right? Hence, the cells are unit of life, because all the metabolic activities are actually performed or carried out by the cells only, right? So this is why we are calling the cells as the unit of life or the fundamental unit of life. So this is the complete solution of question number 10. Now let's discuss next question, that is question number 11. What are the nuclear pores and state their functions? So what are the nuclear pores? Nuclear pores are the tiny holes present in the nuclear membrane. Nuclear pores are the tiny pores present in the nuclear membrane, and these holes or nuclear pores are actually formed by the fusion of two nuclear membranes, right? Nuclear pores are the tiny holes present in the nuclear membrane which is formed from the fusion of two nuclear membranes. Now what are the functions of these holes or these nuclear pores? These holes or nuclear pores allow some material to be transported within the cell or within the cytoplasm from the nucleus, right? They allow some materials or some substances such as RNA and protein in both the directions, right? So this is the function of nuclear pores or holes. Now let's write answer of this question number 11. Nuclear pores. Nuclear pores are the tiny holes present in the nuclear membrane. Which is formed by the fusion of two nuclear membrane, Now, if you talk about the function of nuclear pores, then these holes or nuclear pores. Nuclear pores. These holes or nuclear pores allows the transportation of some substances within the cell or cytoplasm or to the cytoplasm from it, right? From it. It allows some substances or molecule such as RNA and proteins, in both the direction for the process of transcription and translation, right? So it allows some substances or molecules such as RNA or proteins in both the directions in the cell, right? So this is the function of nuclear pore or holes. So this is the complete solution of question number 11. Now let's discuss the next question, that is question number 12. In this question, both lysosomes and vacuoles are endomembrane structures, yet they are differ in terms of their function. That means the function of lysosome and vacuoles are different. So we have to comment on this, that both lysosome and vacuoles are endomembrane structures, but they differ in their functions. Why? So first of all, what is lysosome? Lysosome is actually the membrane bound vesicular structure, right? The lysosomes are membrane-bound vesicular structures holding a variety of enzymes such as lipases, proteases, carbohydrases. Right? Lysosomes are the membrane-bound vesicular structures holding a variety of enzymes like lipases, proteases, carbohydrases. Now, lysosome plays a very important role in the digestion of worn out cells, right? So the function, or they actually involve in the function of digestion of worn out cells, right? Also, they play a very important role in self-digestion of cells. That is why we are calling the lysosomes as suicidal bags, because they are involved in the digestion of cells. They're involved in the self-digestion of cells, right? That is why we are calling them as suicidal bags, right? Now, if we talk about the vacuoles, on the other hand, vacuoles are the storage sacs found in the cells, right? They might store some metabolic waste of cells. In case of unicellular organism, the food vacuole contains or holds food particles. Also, vacuoles helps in the expelling out of excess water and waste from the cell. So this is what the difference in the functions of lysosome and vacuoles we have. So let's write the answer of question number 12. Answer number 12. Lysosomes are the membrane bound vesicular structures. Lysosomes are the membrane bound vesicular structures involving or holding a variety of enzymes like lipases, then proteases, and carbohydrases. Right? So lysosomes are the membrane-bound vesicular structures which are holding a variety of enzymes like lipases, proteases and carbohydrases. These are involved in the digestion of worn out cells. They also known as suicidal bags. They are also known as suicidal bags. They are also known as suicidal bags because they are involved in self-digestion of cells. They also digest foreign particles. That is why they are called as waste disposal system of cell. Waste disposal system of cell, right? On the other hand, if we talk about vacuoles, then on the other hand, the vacuoles are storage sacs found in the cells, right? They might store waste of cell. But in case of unicellular organism, the food vacuole contains food particles in it. Right? So, also this vacuole, these vacuoles helps in the expelling of excess water and waste from the cell, right? So this is how vacuoles and lysosomes differ in function, even though they too are endomembranous structures. So this is the complete solution of question number 12. Now we are going to discuss the second last question of this chapter, that is question number 13. Describe the structure of the following with the help of labeled diagrams, nucleus and centrosome. So we have to discuss the detailed structure of nucleus as well as centrosome along with the labeled diagram of these two. So first of all, we are going to discuss nucleus. What is nucleus? Right. So nucleus is a structure which controls all the cellular activities of the cell. Right. Nucleus is the cell organelle, of course, which controls all the cellular activities of the cell, which is actually composed of various kind of structures such as nuclear membrane, nuclear matrix or nucleoplasm, nucleolus and chromatin. right? All these are the structures or components which are going to make up a nucleus, which controls all the cellular activities of the cell. Now let's discuss this in detail. What are these structures which are composing or which are forming the nucleus? So first we have this nuclear membrane, right? Nuclear membrane, if you talk about, this is the membrane of nucleus. This is the membrane of nucleus. First of all, the nucleus is a double membrane bound structure, right? So nuclear membrane are the two membranes enclosing the nucleus, right? The space between the two membranes of a nucleus is known as perinuclear space, and this nuclear membrane is actually separating the content of nucleus from the cytoplasm of a cell, right? This is separating the content of nucleus from the cytoplasm of a cell. It is 2 in number, and the space between two membranes of nucleus is known as perinuclear space. There are also holes or nuclear pores present in the nuclear membrane, which allows the transport of some substances in and out of the nucleus. So this is about nuclear membrane. Now let's come towards the next component or next structure of nucleus, that is nucleoplasm or nuclear matrix. So nuclear matrix or nucleoplasm is the homogeneous granular fluid which contains chromatin and nucleolus. Now what is nucleolus? That is the third component or the third structure of nucleus. Nucleolus is the region inside the nucleus which is not bound any membrane, which is not bound by any membrane, and is rich in the RNA and protein content, right? This is also, nucleus is also a site for the formation of ribosomes. This is nucleolus. Then the last structure or last component of nucleus we have is chromatin. What is chromatin? Chromatin are the entangled mass. Chromatin is the entangled mass of thread-like structures, right, which is present or embedded in the nucleoplasm or nuclear matrix. This chromatin contains DNA and a basic protein known as histone. So this is the detailed study of the structure of nucleus, and we are going to make the labeled diagram of nucleus as well. Now let's come towards the detailed structure of centrosome. What is centrosome? Centrosome is a structure which is made up of two centrioles. Right. So centrosome is actually composed of or made up of two centrioles which are lying perpendicular to each other, right? These centrosomes are made up of two centrioles which are lying perpendicular to each other. Each centriole is having a cartwheel organization. Each centriole which are lying perpendicular to each other, which are making centrosome, are having cartwheel organization. Now a centriole is made up of microtubule triplets. Both the centrioles are made up of microtubule triplets. Now the proteinaceous hub is present. There is a proteinaceous hub present in the central part of the centriole, right? Now this central part, which is proteinaceous, is actually connected or linked with the help of radial spokes to the triplet of microtubules, right? Now these centrioles help in organizing the astral rays or spindle fibers during the cell division. Also these centrioles help in the formation of basal part of cilia and flagella. So this is the detailed study of centrosome, and we are going to make the labeled diagram of centrosome as well. So let's start writing the answer number 13. So first we are going to talk about nucleus, right? So what is nucleus? It is the structure which controls all the activities of cell, right? Now this is made up of various structures. Various structures. Like the first one we have is nuclear membrane. Nuclear membrane. What is nuclear membrane? This is a membrane. This is a membrane which separates the content of nucleus, the content of nucleus from cytoplasm, from cytoplasm. The space between two membranes, two nuclear membranes, is perinuclear space, is perinuclear space. Also, the very important thing, that is having nuclear pores or nuclear holes in them, right? So, nuclear membrane also consists of nuclear pores which allows the transportation of some substances The next is we have nuclear matrix or nucleoplasm. Nuclear matrix or nucleoplasm. This is the homogeneous granular fluid which contains nucleolus and chromatin. Right? The third component or third structure which makes up the nucleus is nucleolus. What is nucleolus? This is the region. This is the region which is not bound by any membrane. Which is not bound by any membrane and is rich in RNA and proteins, and is the site of ribosome formation. The last structure which makes up the nucleus is chromatin. Now what is chromatin? This is the entangled mass of thread-like structures. These are containing DNA and a basic protein known as histone, right? So this is, we know about nucleus. Now we are going to make a labeled diagram of nucleus on the next slide, that is here. Let's make the labeled diagram of nucleus over here. So this is the outer membrane of nucleus, and these are the pores. We know that the nucleus is a double membrane bound structure. So this is a double membrane bound structure. Here is the nucleolus, which is not bound by any membrane. This is nucleolus, and there are entangled mass of chromatin material, a thread-like structure. So these are nuclear pores. Then these are the membranes, nuclear membranes. This is chromatin. Then we have here is nucleolus, right? Now this is the diagrammatic representation of nucleus. Now we are going to write the explanation or detailed study of a structure known as centrosome. So let's write about centrosome. Here we are going to write about centrosome. So these are made up of two centrioles. These are made up of two centrioles lying perpendicular to each other. They are lying perpendicular to each other, right? Each centriole have cartwheel organization, right? Now a centriole, whenever we are talking about centriole, first of all, centrosome is made up of two centrioles which are lying to each other. The centrioles, two centrioles are lying or located perpendicular to each other. Each centriole have a cartwheel organization. Now we are talking about the structure of centriole. A centriole is made up of microtubule triplets. The proteinaceous part of central of centriole is connected to these triplets with the help of radial spokes. These triplet of microtubules are connected to the proteinaceous central part of centriole with the radial spokes, right? Now, if we talk about the function of centrosome or centriole, then they help in organizing the spindle fibers, they help in organizing spindle fibers and astral rays, they help in the organizing spindle fibers and astral rays during cell division. During cell division. Also they form the basal part of filament and cilia. So these are the functions of centrosome, right? Because the centrosome is made up of centriole. The function of centriole is this, right? Now let's make a diagram of centrosome. So this is the cell, dividing cell. Here is the centrosome. Here is also a centrosome. We know that centrosome is made up of two centrioles which are lying perpendicular to each other like this. They are lying perpendicular to each other. Now they divide, these centrioles divide the spindle fibers or astral rays during the cell division like this. Suppose these are the chromosomes. Right, these are the chromosomes. They are dividing the chromosomes like this. So these are the centrosomes, right? These are the centrosomes, and these two which are lying perpendicular to each other are centrioles. These are centrioles, right? So now we have completed the solution of this question number 13. Now moving on to the last question of this session or of this chapter. This is question number 14, and this question number 14 is asking what is a centromere. So we have to tell what is centromere. Also in this question we have asked that how does the position of a centromere forms the basis of classification of chromosomes. Support your answer with the diagram showing the position of centromere on different types of chromosomes. Alright, so first of all we have to answer what is centromere. So what is centromere? Centromere is the primary constriction on the chromosome. It is nothing but a primary constriction present on the chromosome where the chromatids of a chromosomes are held together, right? Centromere is the primary constriction present on the chromosome where the chromatids of a chromosomes are held together. Now, on the basis of the position of the centromere on the chromosome, we have basically four types of chromosomes. That is metacentric, submetacentric, acrocentric, and telocentric chromosome. Right? So we have four types of chromosomes based on the position of centromere on the chromosome. Now let's discuss each of them one by one in detail. The first one we have is metacentric chromosome. Now in this type of chromosome or in this metacentric chromosomes, the centromere are present at the middle of the chromosome, right? The centromere is actually present on the middle of the chromosome and dividing the chromosome into equal arms, right? The next one we have is submetacentric chromosome. In the submetacentric chromosome, the centromere is situated slightly away from the center or middle portion of the chromosome. These type of chromosomes are termed as submetacentric chromosomes, right? And which is resulting into the formation of one long arm of chromatid and one short arm of chromatid. The third type of chromosome we have is acrocentric chromosome. In this type of chromosome, the centromere is situated near the terminal of chromatid, near the terminal of arm, right? And resulting in the formation of very short arm and a very long arm. The next and the last type of chromosome we have is telocentric chromosome. In these type of chromosomes, the centromeres are present on the terminal of the chromatids, So these are the four types of chromosomes based on the location of centromere in the chromosome. Now let's write the answer of this question. That is answer number 14. Right, so what is centromere? First of all, we are going to write centromere. So centromere is the primary constriction, is the primary constriction present on the chromosome where the chromatids are held together. Where the chromatids are held together. Now, on the basis of position of centromere, we have 4 types of chromosomes. 4 types of chromosomes. The first one we have is metacentric. Metacentric. Right? On the basis of position of centromere present on the chromosome, we have 4 types of chromosome. The first one we have is metacentric. So what is metacentric chromosomes? The centromere in these type of chromosomes, are situated in the center, right? And this is how they look. This is the centromere and these are the chromatids, resulting into the equal arms of chromatids, right? So this is the metacentric chromosome. The next one we have to discuss is submetacentric chromosomes. So now what are the submetacentric chromosomes? In these the centromere are situated slightly away from the center, resulting in one long and one short arm of chromatid, and this is how they look. If we make the diagram, then this is the short arm of chromosome, this is the centromere, and this is the long arm of chromosomes, right? The centromere is slightly away from the center of the chromosome, resulting in the formation of 2 chromatid or 2 arms, which is one is long and one is short. Now, if we talk about the next, that is third type of chromosome we have is acrocentric chromosomes. Now, in these chromosomes, the centromere is situated near the terminal of chromatid arm, right? So, in these chromosomes, the centromere is situated near the terminal or chromatid, resulting in one very short arm and one very long arm. And if we make diagram, then they are going to look like this. Here is the very short arm, here is the centromere and a very long arm of chromatid. So this is how they look, acrocentric chromosome. The last type of chromosome we have is telocentric. Telocentric chromosomes. Now, the centromeres in these chromosomes are situated at the terminal, right? The difference between acrocentric and telocentric is the centromere in acrocentric chromosome are situated near the terminal, whereas in telocentric chromosomes the centromere is situated at the terminal or and of the chromatid. On the chromosome. And if we make a diagram of telocentric chromosome, then they are going to look like this. One arm is not going to show you. This is how a telocentric chromosome will look like, right? So with this we have completed the solution of last question of this chapter, and with this we are ending this session. We have completed all the NCERT questions of the chapter Cell, the Unit of Life. This is all for today. Thank you and have a nice day.